needing around 15 to 20 minutes of your class extra time, that is. So the class yeah. Fine. So let me start with the class. So when it comes to mutations and mutagens, basically you get to see that a mutation is something which actually, a chemical change in which, the structure of the DNA will be changed. So, and this, if at all it is not getting repaired, it will go on, resulting in a change in the DNA sequence. So this is known as a mutation. So mutation is the failure whenever there is no DNA repair taking place. So development of the organism is basically because of DNA repairing and because of DNA is basically because of which any kind of evolutionary change, if it's taking place, it is owing to the stability of the DNA as well. That has to be maintained because of the because of the continual replication of the DNA that is taking place. So any particular point wherever there is any kind of change in that particular, what do you call it, a DNA sequence or the protein sequence results with a completely, undesired sequence or consequences basically. So if you see, living cells have to have some kind of a mechanism wherein even if there is any kind of involvement of mutation taking place, you have to have a repair mechanism so that this can have this will not cause much of a damage. So basic, if you come to it, you get to see that genes are made up of DNA. So the chemical structure is a nucleotide and a nucleoside. So that is sugar and a phosphate. And in the same case, that's what nucleotide and nucleoside is the same thing basically, with only sugar, phosphate, and the third part is a base that is involved. So there are 4 different bases. We have already seen that: adenine, thymine, guanine, and cytosine, with the letters A, T, G, and C. So DNA is made up of 4 nucleotides, that is each having a base of adenine, thymine, guanine, and cytosine. It's called A, B, C, D. And a set of 3 nucleotides, that is a trinucleotide that is there. The string of nucleotides, but again a small part of a string is you have 3 nucleotides as base pairs or base and these other they, after getting transcripted, DNA transcription takes place, then translation. So whenever transcription and translation takes place, you have the codon forming, and then you have the genetic code that is taking place because of it. Again, over here, when it comes to that, we have mutations which actually play a role. Just like so, the different kinds of damages that can take place because of this include, DNA gets damaged by either physical, chemical, or environmental agents. Can be classified into broadly 4 different categories. That is single base pair alteration, basis alteration, that is 2 base alterations, chain breaks, and cross linkages. Again, For this, importance is, whenever you're reading the genetic code, that is when, the importance of all of this comes into place basically. So first step is either production of the genetic code, and then you have the translation of it. Production of the DNA, and then after the replication of the DNA takes place, that is the transcription part, you have the translation. That is the in transcription you have DNA giving rise to mRNA. Then in translation, that mRNA is giving rise to the proteins. So all these codons that are present as the three trinucleotides, they are transcripted into mRNA. They code for three different kinds of mRNA for that matter, codons, and this will give rise to a protein. So many such nucleotides together, they get translated to form the protein. So this is done in a specific order, which is start and a stop. So if at all there is any kind of mutation taking place, it will take place in this particular in the genetic sequence as such. So the different types of mutations, as you can see, are single base, two base, chain mutations, and the cross linkages, damages that can take place. So in this you can again see deamination is amino group is not added. Basically, it gets removed from that particular cytosine, so that you have uracil formation, and then you have spontaneous depurination, wherein cleavage of glycosyl bond is taking place, and then depurinated sites are also called as abasic sites. So again, just a second. We will come to this in some time. So let us see the different kinds of mutations. So change in DNA structure because of either chemicals or radiation or anything for that matter is known as mutation. And the substances that cause this is known as a mutagen. So changes occur in the DNA so that it is reflected in the replication itself, transcription, and translation as well. Again, two major types. That is, if you get to see, it is point mutation and friendship mutation. Point mutations are in what happens is replacement of one base pair by another base pair results in point mutation. So two subtypes again. So purine or pyrimidine is replaced by another. Transversion is wherein purine is replaced by pyrimidine or vice versa. So this is how point and friendship mutations take place. This is transition wherein purine is replaced by pyrimidine over here. You see. So instead of coding for a serine, there is going to be a different kind of amino acid that is being coming all together into place. So you have silent mutations also, and you have missense and nonsense mutations that take place. So in the case of single base substitution, like I told you, point mutation, transition and transversion are the two types. And then you have missense mutation, wherein what happens is nucleotide alters the codon so that the amino acid that is produced is not the same that has to come. For example, in the case of sickle cell anemia or disease, what happens is adenine is replaced by thymine at the 17th position, because of which what happens is instead of glutamic acid, you get valine. It starts encoding for valine. So this sixth amino acid becomes valine instead of glutamic acid. This results in the sickle cell disease. Now, when it comes to nonsense mutation, that's what. And in the case of nonsense mutation, what happens is base is altered. That example with the figure is given in your textbook. You can just have a look at it. So what happens is production of a stop codon takes place. So this produces a stop signal. So what happens is protein synthesis is stopped altogether. So in the case of silent mutations, what happens is codon containing the changed base may code for same amino acid. So UCA has, is for serine. So even if the third amino acid that is, gets changed, even if third base gets changed, it will code for serine. This is again because of the degeneracy of the genetic code. So it is not exactly determined or detected until and unless you do the genetic sequencing. So this is about the different kinds of silent mutations. When it comes to the splice site mutations, what happens is introns are removed and during the translation. So mRNA only has the exon part to it. So introns which have been removed as a mRNA, they have to be done with the particular with a lot of precision, I mean. So nucleotide signals at the splice sites guide this enzymatic machinery. So if any kind of mutation alters, what happens is intron does not get removed, gets or is actually remaining as a part of the final mRNA molecule or the RNA molecule. Translation alters the protein product that is going to come out. So we also have indels and the frameshift mutations and the duplications and translocations. We've already just kind of had a look at it. We will just again look at it again. So in frameshift mutations, you have the insertion deletions. So what happens is because of which either one codon insertion of a single nucleotide gets added, because of which the sequence changes, because of which a different protein product altogether comes off. In case a deletion of a nucleotide sequence or a nucleotide occurs, because of which again the reading frame will shift, because of which again a undesired or a different protein product comes out. So this is taking place. In the case of duplications, what happens is doubling of the genome takes place, because of which you have paralogous genes that are arising. This has been adaptive basically. Now in the case of translocations, what happens is there is a break in between the two non-homologous chromosomes. This results in the gene function not taking place at all. This kind of translocation has been seen in Burkitt's lymphoma and in the Philadelphia chromosome, because it is seen in the leukemic cells of the chronic myelogenous leukemia. So it's known as the Philadelphia chromosome basically, because it is a result of the translocation of the chromosome part. So this produces a compound gene which is known as BERABL. This is present in the chronic myeloid leukemia patients basically. So when it comes to the mutations, the effect of it has been seen basically. So effect of the mutations on the gene product, as you can see, consequences of this, you get to see what happens is that you have the copies should be made, both the copies should be made, that is XX and XYs have to be made in the case of the both male and the female. So if at all there is any faulty copy in this chromosomes, this causes issues, obviously. So this leads to the production of incorrect protein, because of which either there is reduced amount of protein, no protein produced, or if there is some kind of a damage. So this impact of the gene, mutation on the autosomes in the males and the females can usually be the same. Example is autosome 1 to 22. Usually it's the same. So again, there are different kinds of mutations that are seen, recessive mutations and dominant mutations. When it comes to recessive mutations, that is both the parents should have the faulty carrier gene. So both the parents should be carrier. That is when the gene will be expressed. That is, you have an absence of that protein usually. So no gene product or incorrect gene product or incorrect amount is produced in the cells. So this is seen in the recessive mutations. So what happens is, in the case of recessive mutations, the carrier is the risk of a person having a recessive mutation is 25%, because both the parents should be carriers. So it's usually autosomal recessive inheritance that you get to see, and it is either thalassemia or sickle cell anemia. That is what the example of recessive mutations. And also when it comes to dominant mutations, you get to see irrespective of whether both the parents are affected or only one parent is affected, the child usually has that particular condition. So there is a 50% risk because in if both the even in the male for that matter, if it is the X gene which is affected, or if it is the Y gene that is affected, then the male will be carrier. Definitely the Y gene affected, then male is the carrier. His son will definitely be affected. Whereas if it is a daughter, then she will not be affected. But if it is on the X chromosome of the affected father, for that matter, and it is on the autosomes basically. So because of which, what will happen is the affected father will definitely be giving that particular gene which is affected 50%. So what happens is there's a 50% risk or 1 in 2 chance in through every pregnancy that is occurring. So example of this autosomal dominant inheritance is dwarfism, that is achondroplasia, and neurofibromatosis. These two examples are conditions of dominant mutations. Now, when the genes are located on the X chromosome, please just give me a moment. I have another slide that I would like to share with. Right. Again, what? Right. So you have seen the autosomal dominant and autosomal recessive. What it means is the affected father. So there's, in the case of dominant, what happens is you have almost 50% chance. Whereas autosomal recessive inheritance, unless and until both the parents who are carriers, then you have a 25% chance for this to occur. So the different kinds of diseases have been mentioned over here. So when it is genes located on the X chromosome, then again, effect of this mutation is different for males and again different for females. So since males only have, since males have only, one X chromosome and the other is Y chromosome, but females have 2 copies of the X chromosome. What happens is the X chromosome copies in the female is switched off or inactivated. So both males and females have only 1 active X chromosome each. This copy is then activated in both the males and the females. This process is called as the the process by which this X chromosome is switched off is again random. So what happens is X chromosomes coding for the correct product while others have this incorrect or reduced amount of the product or no product at all due to the faulty gene. This is happening in the X chromosome basically, and how it is going to function. When it comes to X-linked recessive mutations, what happens is a female who has a mutation in this gene in one of the X chromosomes, but a correct copy of that other X chromosome is known as a carrier. We all know that. So the X chromosome in the female copy is inactivated. Some of the cells contain the X chromosome in which the correct copy of the gene is also located. Others will have faulty gene. So some cells will have for the correct gene product. Other cells will have information for the faulty product or no product at all. So females who are X-linked genetic carriers usually have only half of the cells having the correct gene information. So what happens is it still will help in the female being normal in nature. The correct protein is being produced still. So the woman will not show any effects. But then the mutation is hidden or recessive when it comes to that, and the female is known as a carrier. So there's a 50% chance of the woman giving this faulty eggs to the children. So 50% chance for the faulty X to be in the child. If at all that child has the mutated X or faulty X or the recessive X, then the chance of the daughter having the same carrier gene is around 1 into 2, that is 50% chance. Again, same thing even for the males basically. So the process in which the X chromosome is switched off is random. So this again leads to or affects the X-linked recessive mutations as such. Now, when it comes to X-linked dominant mutations, what happens is it impairs the cell's ability to make this type of product. So the mutation is known as dominant. Irrespective of both the X's having only one of the X having this kind of mutation, the gene product is going to be affected. The protein, whatever is going to be produced, is going to be affected. So a woman who is X-linked carrier of a dominant mutation has a 50% chance through every pregnancy in passing that recessive X-linked mutation to both her sons and daughters, and they will be affected by this condition because of the faulty X line. Again, despite the woman will be affected because of this disease, but because it is only present on one gene, that mutation, what happens is only one X is affected. So there's a 50% chance for the woman to give that faulty gene to the progeny. This is seen in the X-linked dominant inheritance, and these are quite rare in fact. So when it comes to the frequency of mutations, mutations are not very frequent in nature. They are quite rare. So with the number of billions of bases that are present, the number of genes and the number of DNA, like I told you, what is present, each DNA inside the cell is around 2 meters tall, and we have billions of cells in our body. So the amount of DNA, the amount of transcription, translation, replication that is taking place, if you just imagine it for even a couple of minutes, just looking into it, it is astronomical in nature. So anything mutation occurring also, it is going to be, if it is occurring frequently, then gone case. So in that case scenario, what happens is, it is not exactly occurring very frequently. So estimation is that, there are around occur for every 1 in 50 million nucleotides added to the chains. So that means that in the 6 to 10 base pairs, 6 into 10 base pairs in a human cell that is present, that for every new cell that is adding, you have around 120, only around 120 new mutations. That itself is sufficient. But at the same time, these mutations are stopped at the time of generation itself. They do not go beyond that. You have base pair or DNA base pair, DNA repair mechanisms which take place. If the DNA repair mechanism does not work, that is when this mutation will be carried on to the next replication, and it will, if at that particular point of time also it does not get repaired, that is when it will go on. This continuous, non-repair of the particular cell leads to a different a mutation and a different consequence altogether. So when it comes to that, when should we worry about all of this? So you have to see that around 97% of our DNA does not code for anything. So even if there is any kind of mutation in it, it does not matter. So you have repetitive DNA like ALU elements, you have junk DNA. So what happens is, firstly, if at all the mutation is new, then you have to see the kind of frequency in the particular mutation, and not because the gene is susceptible, because how it is passed by. Then you have got to see if it is dominant or recessive in nature, and then you have to see whether it is autosomal dominant or autosomal recessive, or X-linked dominant or recessive. X-linked dominant is very, very, very rare. And if it is X-linked recessive, that's fine. Again, at least for some, you can deal with it. So some examples of autosomal dominant are retinoblastoma gene is about 8 per million. and osteogenesis imperfecta. So this is one thing wherein, collagen is not exactly encoded properly. And then you have X-linked recessive genes, which is hemophilia, which is around 3 into 10 to the power of 5. So for every 3 into 10 to the power of 5 genes, that is there is a chance for it to occur. And then you have the Duchenne muscular dystrophy, that is around 8 into 10 to the power of 5, wherein the dystrophin gene is affected. So this is the kind of frequency of the genetic mutation that you're looking at. And again, in your textbook, they have given page number 128. You can just have a look at it. Different organisms, genome sizes, and what is the kind of mutation per the genome per every replication. And if you see, the chance of it is point not, and then you have. So it is around 1 in 1,000, 1 in 10,000, 1 in 1 lakh mutation that is occurring for the kind of genome that is there, which is very, very, very less. Again, the contribution of males and females in the mutations, if you get to see. So most of the mutations take place in the S phase of the cell division. So please give me a moment. Right. So I'll just like resume my sharing of this particular PPT. In fact, so when it comes to the contribution of males and females in the case of mutations, you get to see that since it occurs mostly in the S phase, around 24 mitotic divisions occur after the fertilization takes place. So that the embryonic development of the little girl's embryo and then setting of the future egg is taking place even before the baby is born. Whereas the sperm of the 30 years old man, in contrast, what happens is the descendant of at least 400 mitotic divisions since the time fertilization takes place. So fathers are more likely than mothers to transmit any kind of mutations that occur because of chromosomal aberrations, that is aneuploidy. So what happens is that is more apt in the eggs than in the sperm. So children of aged fathers suffer more genetic disorders than that of the young fathers because of more amount of mutations that can be seen. So when it comes to this, it is known as a male bias. So it's not as bad as the number actually suggests. This is because again, mutations occur independently of the DNA replication so that it would affect both males and the females equally. Now, even in the older man, fresh sperm, whatever is coming, that is being held in the, it has been held in reserve, but it is not the, result of years of mitotic divisions per se. And evolutionary mechanisms, you have the DNA repair mechanisms that come so that, sperm gets repaired as such. So when it comes to the types of mutations that occur, you have somatic and germline mutations. Significance is that mutations that occur in the somatic cells, that is bone marrow, liver, example, they damage the cell. They either make the cell cancerous or they kill the cell altogether. Germline mutation, what happens is it affects the progeny that is going to come. Simple. So you have a mutation test that can be done, which is the Ames test. I was looking into it actually. The thing was, I was looking whether, we have the Ames test per se with, do I have a picture of it or not, in fact. So just give me 1 minute. I'll look in that. So in the case of Ames test, what happens is, what you can do is, in this, it was developed by Bruce Ames. So for that matter, what happens is the bacteria is mutagenic in nature, that is cancer causing. So what happens is you use Salmonella typhimurium for Ames test. And then that is going to be unable to synthesize the amino acid histidine. Okay. And this, if I'll just show you the image. Right. I hope you can see. So Salmonella strain is taking place. You have a required histidine in this. Then you have a possible mutagen. Then you plate it with minimal histidine and incubate. So after this, what happens is in the histidine positive to histidine negative strains, you get to see that the mutagen has caused mutations. So this is what is being seen. More number of colonies and less number of colonies. So a suspension of this histidine strain of Salmonella has been plated with a mixture of rat liver and with an agar lacking histidine. Then around aminofluorine, which is around, carcinogen is added. Then it causes mutagenic effect on these two plates. Then you have colonies forming. So with minimal histidine, and you see there are less, more number of colonies over here. That is, many chemicals are not mutagenic but can be converted into mutagens. So what happens is, in this Ames test, a positive one, you get less. This is the control plate, which natural revertates. Over here, you do not have colonies, So there's a lot of mutated, colonies over here. So examples of chemicals that are used and which are mutagens are ethylidene bromide, dibromide. Then you have, it is added to gasoline, and then you have Zeram, which is used to prevent fungal diseases, and you have isoniazid, and then you have different kinds of food additives also, which are again mutagens in nature. So it has been banned. You have saccharin, which is some kind of a carcinogen. It does not give a positive Ames test. So some aflatoxins, and they again are mutagenic in nature. So Salmonella typhimurium is a prokaryote. It's not exactly a perfect model. So that's why you add the liver enzymes that is present over here, that the rat liver extract as such. So this is all about the Ames test. When it comes to the transgenic animals developed for detecting the mutagens, you have quite a number of them that have been developed right now. Big Bloom mouse, which has one LAC-I gene, that is the lactose operon repressor gene as the target gene. Then Muta mouse, and some other animals that have been developed with the streptomycin sensitivity, or the delta mouse having the xanthine guanine phosphoribosyl transferase gene. So they were developed for testing this mutagenicity of the products, but again useful tools for determining how much of mutagenicity is there in the various chemicals in the environment. So you have zebrafish and Medaka also, which are actually used have been developed for testing the amount of mutagens in the chemicals in aquatic environment. So again, now you will ask me one question, that is, if at all if all the mutagens are harmful in nature, or whether there is anything which actually provides any kind of is there any beneficial mutation as such? So when you get to see that, when in case of beneficial mutations, and in the case of you get to see that antibiotic resistance is developed in certain bacteria, because they're easy to study also. It is you can actually see. And whereas what happens is, some bacteria are present because of any kind of genetic mutation. What happens is, they get to eat plastic or nylon, for that matter. Short molecules of nylon oligomers have been seen after mutation. The bacteria start eating it up or breaking it up, basically. So this is because of a friendship mutation the bacteria have undergone. And then you have sickle cell resistance to malaria. I could not exactly find any kind of PowerPoint or information regarding these. So you'll just have to bear with me and listen to the class. Please look into your textbooks if you can. That should be helpful. Page 132. So sickle cell resistance to malaria also. So because of mutations, what can happen is it decreases the normal efficiency of the body. So it's quite a bit advantageous. Lactose intolerance in adult mammals, certain people do not have lactose tolerance. Whatever, milk or milk products, whatever they have for that matter, the gut is not able to digest that. So what happens is, in this in this particular example, that lactose tolerance mutation appeared in a group where it was advantageous for them. So what happens is, there was a genetic drifting in which, a few people within the group, they discovered that they could start consuming milk also. Resistance to atherosclerosis. This is one mutation that has been favorable, because of which, the original mutation has been identified because of the resistance and the mutation, basically. And people have lived longer, and diet and lifestyle habits, have changed, because of which it has become advantageous to the people having it. Now, the most surprising and beautiful example is immunity to HIV. So there are certain people who are immune to HIV as such, because HIV cells affect the T lymphocytes, macrophages, dendritic cells, and neurons. So AIDS is when all these cells are killed off, so that the person dies because of other infections rather than because of AIDS. So HIV virus molecule attaches to the surface of the T cells. So that is CD4 cells, basically CD4 receptor or the CC chemokine receptor 5. So what happens is, some people have a mutant allele of the CCR5 gene. So this lacks the expression of this protein on the surface of the T cells. So HIV virus cannot attach itself to this particular mutated CCR5 allele, because of which homozygous individuals are resistant to HIV infection and AIDS. And this frequency is quite high in some populations that have never been exposed to AIDS, so that it is a prior selection for this particular allele. Now, so this is quite interesting, in fact, whatever we have read so far. Now, if you see the mutations in case of the fruit fly, that is Drosophila melanogaster, you see that normal or the wild type have normal length, normal shape of the wings. But genetic mutations alters this. So you have shortened wings or vestigial wings that does not allow them to fly at all. So it is because of a recessive gene. Now, in this, what happens if only one gene is mutated, healthy version can dominate or will stop the mutated effect, so that this defect is overridden. Some flies also have curled wings. That is because of curly gene. So all of this is because this is there on the second chromosome. Again, it's a dominant mutation. So if both the copies are mutated, then the flies don't survive in the first place. So this is all about the good examples or the benefits of mutation. Now let us see what exactly are mutagens. Just give me a moment, please. Right. So mutagen is a natural or a homemade, what do you call it, or a human-made element, basically, which causes, I'm sharing my screen. Just a second. They change the genetic information of an organism, usually by changing the DNA. So they are also known as carcinogens because they cause cancer. So one of the first report of this is with the use of nitrogen mustard or the mustard gas during World War I. So this has caused a lot of mutations. And so because based on the mode of action and the kind of mutagenicity, you can actually identify and classify these mutagens. So let us just see. So this is the basic structure of DNA, like I told, sugar, phosphate. Then so you have the phosphodiester bond. So you have gene mutations and chromosome mutations taking place because of it. So the thing is, getting an appropriate, what do you call it? Getting an appropriate slide is also important. Not exactly happening all the time, basically. get this particular. Yes. Please give me a moment. Skip. So anyway, let me continue with my lesson as such. So what happens is in the mutagen, you have different kinds of mutagens again. You have base analogs, then you have, what do you call it, chemicals that actually alter the structure of this particular right. So they alter the structure and the pairing properties of the bases. Then you have intercalating agents, and you have agents that alter the DNA structure altogether. So when it comes to that, you get to see that when it comes to base analogs, what happens is they resemble the purines and the pyrimidines. So in the place of the normal purines and pyrimides, they go and they start working. So example is bromouracil. So it's an artificially created compound that's present in the research basically. So it has a high likelihood for tautomerization. So as to form the enol form, that is BU, that is bromouracil form basically. Instead of, it resembles the thymine. Uracil resembles thymine. So that's what. So the bromine atom, instead of the methyl group, it's present over there, and this will be incorporated into the DNA. In the case of aminopurines, adenine analogs, that is, they pair with either T or sometimes C, they causes a T to GC ratio to change or transitions to occur in that. So they cause transitions because of which spontaneous tautomerization occurs. Now, there are certain chemicals that alter the structure and the pairing properties of the bases. Example is nitrous acid and nitrosoguanidine, or methyl methanosulfonate, or you have ethyl methanosulfonate. So all these, for example, nitric acid, digestion of nitrites causes this. So what happens is you have sequences, that is hypoxanthine and the DNA pairs with C causes this transition. So you have deamination occurring. And then in the nitrosoguanidine, like methyl methanosulfonate, or you have ethyl methanosulfonate, they're all chemical mutagens. They react with bases, add methyl or ethyl groups, because of which this alkylated base will get affected, can get degraded to yield a baseless site altogether. So it has a mutagenic, recombinogenic, or mismatch occurring over here in the case of DNA replication. And then you have the intercalating agents, wherein acridine orange, proflavin, ethidium bromide, they're all used in your labs. Acridine orange is a dye. Ethidium bromide is used in the, what do you call it, gel electrophoresis for that matter, for identification of the DNA. So they're all ring molecules. They interact with the bases of the DNA. They cause insertion, causes a stretching of the DNA duplet, because of which what happens is this causes frame shift mutations. Now you have again agents that alter the DNA structure. So what happens is a large variety of these agents are seen. So you have large molecules that can be non-coding for that matter. Then you have agents which cause intra and interstrand cross-linkings. They are known as psoralins. They're found in some vegetables and in treatment for certain skin conditions. And then you have chemicals that cause a DNA strand break, that is peroxides. So they cause mutations, but by induction of the repair genes, this can be reverted back. Then you have radiation. Radiation is one particular mutagen whose effects were discovered quite some time back itself. So Roentgen was the scientist who had actually discovered X-rays. Becquerel had discovered radioactivity. Marie and Marie Curie discovered the radioactive elements. So these three had given the rise to the radioactive and the radiation and the study of it basically. But then What we have in the body also, the X-rays can cause certain mutations. You have the EM spectrum, that is the visible light, and all that other types of the electromagnetic spectrum, right from gamma rays, alpha, TV, what is there basically. And then you have your microwaves. So certain portion of it, any other than the visible light can cause mutations for that matter. For that matter. And ionizing radiation, that is X-rays, gamma rays, they are quite energetic, in fact, and they react with the biological molecules. So you have ionizing radiation because of which, and they also include a term which is known as corpuscular radiation, that is atomic and subatomic particles. They are again alpha and beta particles. They react with the nuclei and the DNA basically. So UV radiation is not ionizing as such, but it reacts with the DNA. So it's a mutagen as well. And the different sources of radiation, you've already seen that exposure to different kinds of diagnostic tests, X-rays, and nuclear tests, nesting and testing in the power plants. You have TV, smoke detectors, airport X-rays. All of it are sources of radiation. And what is the biological effect of radiation? Again, damage to the cells, primarily because of the production of free radicals, that is the hydroxyl radicals. These possess this unpaired electrons, chemically very reactive. They interact with the DNA, proteins, lipids in the cell membrane. So because of which damage takes place. So this causes a blockage in the cell division or cell death altogether. Then the amount of dosage that can be detected with, and after the exposure to radiation is of different types: sublethal, lethal, supralethal. So when there was this atomic bomb explosion in Hiroshima, Nagasaki, what happened was, the different kinds of dose, people were exposed to different amounts of radiation. So sublethal dosage cost around 100 to 250 rems. That is, you have around different kinds of effects because of it. Lethal dosage around 350 to 450 rems, because of which death because of blood cell or gastrointestinal failure. Supralethal for dosage, which is around more than 650 REMs. So because of which, definitely death was because of heart or the central nervous system damage. Again, genetic effects of radiation is ionizing radiation causes breaks in the DNA strands. So rearrangement, deletion, chromosome loss, all of this can occur. Damage to the the bases, so mutation. Then cross-linking of the DNA to itself for the protein, so that is also one of the effect of radiation. When it comes to UV light and exposure, what happens is it is less energetic. Therefore, it is not exactly non-ionizing. Wavelength is again different. So UVC is one which causes germicidal, most energetic and lethal. It is not exactly found in the sunlight, but it is absorbed by the ozone layer. You have UVB. I have this at least. So many of them radiation, So what happens is you have thymidine or pyrimidine dimers that are being formed over here, or thymidine dimers, thymine dimers. So you have tanning beds, for example, have this UVA and UVB. So major lethal lesions are because of the pyrimidine dimers, that is produced by UVA and UVB and UVC. So what happens is they attach to the pyrimidines in one strand. So you have bulky lesions that are formed. So mutation and chromosome rearrangement will occur. So when it comes to mutagens, and so you have a pyrimidine dimer that is being formed here, thymine dimer. So we have just seen this ionizing radiations and what kind of mutations it causes. different kinds of mutations. This is the aim stress, like I told you. Colony of this reverted salmonella, no growth. Anyway, so when it comes to mutagens and carcinogens, so mutagens causes mutations, carcinogen is cancer causing. Simple, basic definition. So when it comes to DNA repair systems, you get to see that in the DNA repair systems, what happens is basic damage. So basic damage to DNA for that matter. This is being repaired so that it can occur before the replication. So as DNA damage occurs spontaneously, so you have to repair the DNA only. So this is the only macromolecule which is actually repaired by the cell. So you have different kinds of repair mechanisms, which is damage reversal, which is simply that the normal structure is restored without breaking that particular backbone. So one is, like I told you, endogenous and exogenous damage that can occur, causes of the DNA damage, types of DNA damage, again, hydrolytic, oxidative, and DNA strand breaks. Because of radiation also it occurs. So mechanisms, you have direct reversal, wherein that particular base is removed, and then you have the damage removal, wherein that particular damaged or inappropriate base is totally removed and from that section of the nucleotides. And damage tolerance, wherein it is the true, it's not exactly truly repaired, but you start coping with it, how it is done. So again, you have different kinds. Before I come to that, you have so damage reversal is in, you have photoreactivation, simplest and oldest repair mechanism. So the pyrimidine dimers, in the case of when it is formed in the UV light because of UVA, UVB, basically this is broken. So photoreactivation is one method. So photolyase is the enzyme that is used. In the case of ligation of the single strand breaks, what happens is X-rays, some certain chemicals like peroxides can cause breaks in the backbone of the DNA. So this is repaired by DNA ligase. This break is ligated together or joined together. So microbial mutants have this high levels of DNA ligase basically, and they are very reactive in nature. And human beings, only 146BR was found in both of the DNA ligase 1 genes. Only one human apparently known by the code name, that is, 46BR. They have not identified or given the identity of that person. So she had poor growth, immunodeficiency, sun sensitivity at the age, and she died very young. So fibroblast cells from this particular lady were sensitive to killing by DNA damaging agents, including the ionizing radiation. And again, there is a rare hereditary disease, that is Bloom syndrome, wherein, you have a DNA ligase deficiency. So there is high levels of chromosome aberrations and spontaneous mutations in such kind of a person. When it comes to DNA damage removal, this is done by the DNA that is damaged, it is removed, replaced with fresh DNA. So example is base excision repair. So in this, what happens is changes in the DNA basis is done. Mechanism is, you have DNA glycolase coming in, glycosylase coming in, removes the base and then clips out the damaged base. You have a gap over here. So DNA repair enzymes come. So in bacteria, it is DNA pol1. In eukaryotes, it is DNA polymerase B. And this, again, there is a chance for proofreading, since it is not taking place, you have enzyme mistake also that can take place. This is how the base excision takes place basically. So DNA glycosylase. So this particular base is removed. So you have an endonuclease that is coming over here. So you have a phosphodiesterase which comes, then pol1 and ligase which comes. So that base is excised. When it comes to nucleotide excision repair, what happens is this distorts the DNA double helix. Then you have that is because if at all there is any kind of damage with the pyrimidine dimer, that is one example of nucleotide excision repair. So whatever bulky damage that is being done to the DNA double helix, that can be removed with the nucleotide excision repair. So what happens is the damaged DNA is there, and it is clipped by an endonuclease on either side of the place where the lesion is there, around 12 to 13 nucleotides apart. Then this DNA which is damaged is removed as a part of a 12 to 13 oligonucleotide base altogether. Then DNA polymerase comes in, fills the gap, and then DNA ligase seals the nick. So 12 to so you have an exonuclease. Then this is the nick over here. Right from here it starts, and this part is excised. Then DNA polymerase and ligase come, and they fill up the gap that is present. And then you have a mismatch repair also, wherein what happens is this failure of normal adenine to thymine cytosol to guanine, if it is not taking place, then this will come into place. Then you have a double strand break repair also, basically either by homologous recombination or non-homologous ending joints together. So they have given this particular example also. When I send this particular slide to you, you can actually have a look at it, how it is going to take place. There's a double-stranded break, so repair complex comes in, BRCA genes come into place. Then you have a recombinant repair also. In this recombinant repair, what happens is you have a replication taking place, strand and exchange. So when the then you have a recombination that is completed over here with a gap. So that is being filled. So in this daughter strand gap which is present is getting filled over here. So example is again BRCA1 and BRCA2. They are involved in this recombination repair together with homologs of the yeast, that is RAD51 and RAD52 for that matter genes. So a second type of recombinational repair is that it is primarily used to repair the broken DNA ends. That is because of the ionization, radiation, or chemical mutagens. This repair system employs B cells and T cells of the immune system for genetic rearrangements. Again, protein kinases are involved for the non-homologous end joining. Then you have the mutagenic repair, that is translation synthesis, which is an alternative scenario, so that this can happen. This actually happens in the bacteria more than that of the eukaryotes. So when we have to summarize this particular lesson, we see that mutations change the normal structure of the DNA. Different kinds of mutations are present. They cause different kinds of changes in the genetic code. They can be missense, nonsense, frame shift, base, then insertion, deletions, duplications, translocations. They are the different types of mutations. Mutagens are those agents that cause the mutations. They can be natural or man-made. Some of the mutagens are carcinogenic, that is, they cause cancer. And frequency is that, depends upon the organism basically. And mutations are necessary for evolution, and so that we get to adapt according to that. So this is the harmful mutations cause an impact on the gene and the product basically, so that if at all the repair mechanisms that are present they fail to correct, then it causes damage. So you have autosomal recessive, dominant, sex-linked disorders, all because of different kinds of mutations. So DNA damage reversal, removal of that particular mutation, or tolerance to the mutation are the three types of repair mechanisms which are seen in the cell. So this is about DNA mutations and DNA repair mechanisms that are there. Now we will start with the unit 6 in our textbook, that is the recombinant DNA technology. I'm going to pause my screen. So in the case of rDNA technology, you get to see that we are going to just see the basic techniques involved in the recombinant DNA technology. Then we are going to see the molecular scalpels, that is the endonucleosis, and what are the techniques in the analysis for the DNA, that is the RFLP analysis. Then we are also going to see the blotting and the vectors which are used for the cloning basically. So when it comes to that, to clone is that you're going to make identical copies of the gene basically. So when it comes to the production of an rDNA molecule, what you're going to see is you're going to have to extract the DNA. So you're going to have to production of this rDNA, production and cloning, or for that matter, any kind of rDNA technology involves a few of the steps. So extraction of the DNA, isolation of the required DNA from the desired organism is supposed to be done. Then this is done by centrifugation technique, so that you get the DNA extract. Then this is added with specific endonucleases, restriction endonucleases, so that you get a palindromic sequence, and this results in the isolation of gene with sticky ends. Then you're going to get a suitable molecule, which is a carrier selection basically. So you're going to use a plasmid for this. Then you're going to go and do the gene splicing. Afterwards, after the gene splicing, you're going to incubate, whatever the isolated plasmids are treated with the restriction endonucleases. Then they are, you get the palindromic sequences. This results in the opening of the two sticky ends. Then you're going to mix them with the DNA ligase. Then you get the complementary ends, to which what happens is, you're going to, they come together with the help of the DNA ligase. This leads to the formation of the rDNA molecule. Then this rDNA molecule has to be inserted into the bacterial cell or the organism of interest. So the produced rDNA molecule is is there in the cell, and the bacterial cells, both of them are suspended in calcium chloride solution, and then the temperature is increased to around 42 degrees Celsius, because of which porosity of the cell wall of the host cell, that is the bacterial cell, takes place, increases, because of which this rDNA molecule enters into the cell. So you have bacterial transformation, and then you have the propagation of it, culture of the transformed bacterial cell. So the cells are again thoroughly screened, transformed cells are taken, and then they are cultured, because of which multiplication of the bacteria with the desired gene is taking place. So this is the basic principle of the schematic, diagram, you can say, of this particular molecular cloning or genetic engineering, basically, or using a recombinant plasmid. So when it comes to the principles of rDNA technology, we have DNA cloning. So they have given you some of the enzymes that are used in the rDNA technology. We will come to this in some time, basically. Please listen to me first. Right. So when it comes to the principles of rDNA technology, you have two main areas. One is DNA cloning, and you have the second one which is DNA analysis. So in DNA cloning, basically what is happening is the desired DNA fragment is amplified. More number of copies of the same DNA is being done. So in DNA cloning techniques, you have the natural in vivo cell-based cloning, that is the DNA replication. And you have the second one, which is polymerase chain reaction. Second is in vivo based cloning. Again, different kinds of, this is again of 6 steps, that is generation of the DNA fragments. I have just read this basically. Then recombination of this particular DNA fragments. Then you have the vectors, which is the carrier of the DNA that is there. So you have that vector which is used, incorporation of the target or the foreign DNA into the vector is taking place. Then you're going to get a recombinant DNA over here. Now different types of vectors are also there, that is plasmids, bacteriophages, cosmids, bacterial, and P1 derived artificial chromosomes, yeast artificial chromosomes as well. So we'll just have a look at them. They have given the same thing in pictorial forms actually, if you get to see. Uses of that, these are the different kinds of enzymes that are used, different kinds of, according to the function. This is the chromosomal DNA. These are the cloning vectors. So in the case of plasmids, you have this plasmid DNAs, the circular DNAs basically, they occur in the bacteria. Most common cloning vectors, around 1 to 200 kilobase pairs long DNA duplexes. So they contain specific genes responsible for antibiotic resistance, can be used to select the bacterial colonies containing the recombinant plasmids. So example is they can actually incorporate up to 10 kilobyte of the foreign gene, but they can also be around 1 to 200 kilobase pairs long. Examples are again plasmid Bolivar Rodriguez 322. PBR 322 is very famous for that matter. And then you have other plasmids such as PSCI 101, that is Stanley and Cohen. Two different scientists had actually developed this particular plasmid. So you have bacteriophage and yeast artificial chromosome vectors also. In the case of bacteriophages, they are also known as phages. They're basically viruses that actually infect the bacteria DNA, usually in the form of a linear duplet, so that it has an overhanging, sticky, or a cohesive end. Let's see if I have a picture of this. So what happens is, in this, it is known as a cos sequence that is present. So this lambda phage is one of the extensively studied and utilized phage basically. It can incorporate up to 10 kilobytes of this foreign DNA. And then P1 bacteriophage also has got large genome and allows up to 100 kilobytes of the foreign DNA inserts into them. And you have cosmids. Cosmids are a kind of plasmid, but it has only the minimum vector DNA necessary for insertion of the largest possible DNA fragment that can be allowed. So they can insert up to 50 kilobytes of the foreign DNA approximately, which is a large amount basically. So this is about the vectors. When it comes to yeast artificial chromosomes, I'm just having that I have yeast artificial chromosome picture. Unfortunately, no. So you please listen to this. So it has minimum DNA sequences that can be allowed for the that is the centromere and the telomere formation, and also the DNA sequences known as the autonomous replication sequences, which are actually required for the replication of the yeast. So yeast artificial chromosomes can actually incorporate up to 1000 kilobytes of the DNA and DNA fragments. This allows the replication of the eukaryotic DNA with repetitive DNA sequences. Then you also have the transformation of the host cells, so that you get competent so what happens is, bacterial cell membrane is actually not permeable to DNA, but it is made permeable by adding certain salts such as calcium chloride, and also high voltage and increase the temperature, because of which the porosity of the cell walls increases. The DNA, the foreign DNA is taken, and then the host bacterial cell will incorporate this DNA molecule with its nucleus and with its DNA basically. So this is known as transformation. Single vector is involved, it is known as then you get a foreign replicate cycle independent of the bacterial cell cycle, so that you get a large identical copies of the target DNA or the clones. So when it comes to the screening of the recombinant vector, you get to see that after many multiplications in the tissue culture after transformation, that is, you can do a recombinant vector screening, so that you can get to see whether your desired genes have been incorporated or not, or inserted into the vector molecule. So you have replica plating for that matter. And then you also have a specific clone selection, wherein different kinds of techniques have been identified, so that you can check for the specific DNA sequences and whether those sequence inserts are present or not. Hybridization of the nucleic acid is one of commonly used technique. So colonies of the transformed host bacteria with the recombinant clones, they are used to make replica plates. Just please give me one moment. Today I would be needing around I would like to continue the class till around 4:45 or so, and try to finish this particular lesson. So please bear with me. Otherwise, we will not exactly be able to finish the syllabus on time. So when it comes to replica plating, you can get to see these are the plasmids that are there. So what happens is restriction endonucleases are added, foreign DNA is also incorporated. DNA ligase helps in joining these particular plasmids all together. Transformation of the cells take place. Selection of the transformation cell. This is by the hybridization of the nucleic acids takes place. So because of which, what happens is you have colonies with the plasmids on the agar-containing tetracycline. Colonies with recombinant plasmids are present and transferred for testing. Again, you can just get to see. So this is one bacterial colony with the recombinant testing that can be seen. When it comes to replica plating, what exactly happens is you have autoradiography, wherein you have transformed bacteria, agar it's plated, bacterial colonies with the recombinant vectors are taken. This is the master plate. Selection of antibiotic resistant colonies or sensitive colonies are both seen. Then after this, you have autoradiography, wherein what happens is DNA of the replica blot is denatured to make the single-stranded DNA that allows to hybridize with a single-stranded radio-labeled DNA or the RNA probes. So, this causes, and then after this causes this particular labeled DNA to be seen in an X-ray film. So this is known as autoradiography or replica plating technique, as it is called as nucleic acid hybridization basically, and autoradiography. This is the steps basically. So in case of a DNA library, to make RDNA, molecules having different sources of DNA fragments can be used. Then what happens is the complementary cDNA is produced, and then you get the collection of this rDNA all together at a place is known as the Recombinant DNA Library, wherein rDNA library of the human genome using the plasmids as a vector will need to have several thousand clones, so that the whole of the genome is being maintained. So you can use yeast artificial chromosomes as the vector with DNA, digested by this restriction enzyme, so that you can get a human genome with around 13 to 14,000 colonies basically. Then you can have cell-free DNA cloning, in which, that is the in vitro PCR reaction, wherein what happens exactly? So DNA sequence amplification is taking place. Then you have this is by the PCR. So it was actually discovered by Carrie Willis and then further developed by other scientists. It is the most reliable tool that is being used in the molecular biology field right now. So what happens in the in PCR reaction is amplification of the DNA segments by a PCR fragment. So you have a primer that is being used basically. So in this, what will happen is, so for the construction of the cDNA library, like I told you, mRNA is isolated, oligoprimers are added, oligo-DT, and then cDNA is constructed using the reverse transcriptase. Then you have the dNTPs present. mRNA is degraded by alkaline hydrolysis, and the dsDNA is prepared using the DNA polymerase 1 and dNTPs. And this is used with the lambda phage vectors or the phage vectors. Around 13 to 14,000 copies are necessary. So when it comes to the cell-free DNA cloning via polymerase chain reaction, you have a primer, which is a short stretch of nucleotides having the complementary sequences. They are attached to a long template molecule. So from the 3' end, that is. So you have so in this method, basically what happens is only 3 steps: denaturation, annealing, and primer extension. So in this, what happens exactly is denaturation because of the heat, that is around 92 to 95 degrees Celsius for around 30 to 60 degrees Celsius is done. Because of it, the DNA double strands break. Before this, what you have to do is you have to give in the polymerase enzymes. You have to add your DNA sample. Then you have to add give the primer sequence basically from wherein your PCR reaction should start. That is 3' end and the 5' direction it has to go in. Then for the amplification and for the amount of DNA that has to be seen and all that, you use the ethylene bromide staining afterwards. So when it comes to the procedure, again, you have DNA strands separated by the heating, that is denaturation. And then you have to cool and add the synthetic oligonucleotide primers that flank this region that has been amplified. So first itself, you'll add the primers, and then you will add the heat stable Taq polymerase, so that this catalyzes the 5' to 3' synthesis of the DNA again. And then you have the annealing part, wherein the process is brought to a lower temperature, so that is around 55 to 60 degrees Celsius for around 30 seconds. Then primers will help in the formation of the C2G and the adenine thymine bonds basically. And this helps in the re-annealing of the original DNA strands also. And then this is all extended, repeats. So you have to set in so that you get thousands and millions of copies. So the number of extension also, number of cycles also you'll have to put in. So after this is done, after one cycle of PCR gets completed, original DNA molecules gets quantitatively doubled. Then this thermal cycle repeats for around 25 to 30 minutes. Each time, desired DNA which is present gets doubled. So in a few hours itself, or in a few cycles, you're going to get millions of copies. So what are the components necessary for PCR? If at all you have done, you might already be knowing. Otherwise, it's the Eppendorf tubes, the template for the DNA, the primers, that is the 3' primer and the 5' primer that you need. You need to have a buffer solution. Then the nucleotides mixture. Then you need Taq polymerase. Then you have to add mineral oil if it is heat sensitive in nature, so as to avoid evaporation. And then you need to have the thermal cycler, which is the machine that actually helps in doubling. So all of it is made up to 100 microliters, and this is going to go on. Now, what is the exact product for the PCR that you're going to get is, you're going to get after the primer extension, you're going to get the PCR product. You have to perform agarose gel electrophoresis, And in this, you know, once you have done, you are going to see the after hybridization, you're going to see short-sized oligonucleotides which are complementary to the PCR produced DNA strands. They are again labeled, and you're going to see with the process of autoradiography, you're going to see basically how it is how it has come. In the case of nested PCR, you're going to get a new primer, which is like, you know, this PCR product is reamplified using a primer product. And in this product, you will get the product which is shorter than the first PCR product. And when you do restriction enzyme mapping, you're going to get the PCR product. And since there is even restriction digestion that is taking place, so you have the restriction enzyme which are like, you know, gained or lost because of the mutations that take place. So again, cloning sequences and all that, they have been given basically. Advantages of PCR, again, it is rapid. You will get the result within a day basically. And small copies of what do you call it, like, you know, the sample is sufficient for that matter. So that, you know, once it is the gene amplification takes place, you can actually see whether the resultant, like, you know, whatever you're looking for is there or not. Example is your COVID virus right now. Basic sample from the nose is taken, or the throat swab is taken with the DNA in it, basically. And whether the presence of a virus is present or not, that is being checked with the help of the PCR, basically. You get the result in one day. So very small amount of the sample is sufficient. But what exactly are the disadvantages of PCR? False positive. You'll get a false positive because if at all there is any kind of contamination, you'll definitely get a false positive result. Otherwise, you'll get a false negative if at all there is any absence, or what you're looking for is again contaminated. So this is one thing. And when it comes to the applications, you get to see it is used in therapeutic, diagnostic purposes, and for even forensic medical investigation, criminology. You can use PCR, and then you can use gel electrophoresis for that matter to check your paternity. So many purposes for PCR. So in situ PCR is one method wherein it is like, you know, DNA or mRNA is amplified by the PCR. Then it uses the DAC polymerase. And then what happens is technical considerations are that, you know, you can see basically direct or in situ gene amplification thing, or indirect in situ gene amplification. I request you all to actually please, you know, bear with me. Another 15 to 20 minutes, we will finish this lesson, because if at all we continue, it is going to be a little bit difficult. Or another 10 minutes up to 3:45, I will take the class and stop. The rest can be done in the next class. So I will be teaching even, what do you call it, up to the northern blotting and the up to DNA sequencing. After that, you can be like, you know, you can leave the class. So when it comes to the ARMS PCR, that is the amplification refractory mutation system, you have, it is a special type of PCR wherein, you know, the primer is a mutant specific. So if at all there is any mutation, that can be identified. So methods, again, you know, in case of DNA analysis, you get to see there are two different, like, you know, different kinds of DNA analysis. It involves nucleic acid probes and nucleic acid hybridization. We will just have a look at it later, because I think we have nucleic acid hybridization over here. We will just have a look at it. Anyway, so this DNA or RNA sequences are labeled radioactive or non-radioactive. They can be used to detect the DNA or the RNA fragments with sequence homology. So mostly they are DNA probes. They can be used for like, you know, different genomic sequence analysis or cDNA sequences, specific genes or oligonucleotides. All of these can be used. So you label the DNA, and then with sites at which the radioactive labeled like, you know, DNA probe hybridized with the complementary DNA sequence on a nitrocellulose filter paper. On a nitrocellulose filter paper, and this is detected by affinity towards streptavidin, which is an like, you know. And then in case of the nucleic acid hybridization, it is done by mixing DNA with two different sources. Then it is denatured by alkali or heat. made to be on a single stranded, and it is allowed complementary base pairing under proper conditions. So you have two different kinds of what do you call it, nucleic acid hybridization. One is northern blotting and one is southern blotting. In the case of southern blotting, it was named after the scientist. So what happens is you have restriction endonucleases that are present in the DNA. Please look into the textbook for you. You can see the picture. It can be used to identify the location of a single gene on any of the chromosomal DNA. So you use the restriction endonucleases, DNA fragments are produced. These are what do you call it? Then the DNA fragments undergo gel electrophoresis. So what happens is separation of the DNA or the restriction fragments based on the size will take place. Then the fragments in the gel are denatured by using alkali. Then it makes it so that you have single stranded genes that are formed. Thank you. And then after this, the electrophoresis of the gel takes place, right? You have a short nitrocellulose or nylon membrane that is used, and the DNA fragments are actually transferred and immobilized onto that. Then the nitrocellulose membrane with this absorbed DNA is treated and incubated with radioactive isotopes. Then again it is labeled with the probes. So it will hybridize with this homologous DNA. It forms radioactive double-stranded DNA homologous DNA probes. This again is located with the help of autoradiography. So this takes place in the, southern blotting basically. Let me pause. So in the case of northern blotting, what exactly happens is, it is used to identify the mRNA now. So RNA is extracted, electrophoresis again is done. So you have the separated RNA, which is based on the size. Then it is transferred onto the RNA, the membrane basically. Then it is heat fixed. So what happens is it is immobilized on the nitrocellulose paper. Then it is separated by the diazotization and with benzyl oxymethyl paper basically. These are hybridized with labeled DNA, which is radio labeled DNA, and this is again seen by autoradiography. So this is the technique that is there in the northern blotting. So just a second, please. In the case of restriction mapping, what exactly happens is, so in this technique, what exactly is happening is, just a second, I did that picture. I thought I did. All right, we'll just continue with it. So in the case of restriction mapping, what exactly happens is that a specific DNA probe is taken. Then again, gel electrophoresis is done. And then you're going to get southern blots of the DNA. And then once you get that, what happens is it is again digested. This is used in combination, gives a detailed mapping of the DNA fragment that is present. Then this is actually called as a restriction map of that particular DNA basically. So You have that in your textbook. So when it comes to DNA sequencing, you have different kinds of methods to sequence the DNA, that is by cloning and amplification of the target DNA fragment, so that the nucleotide sequences are determined in this particular method. So you have dideoxy chain determination method, which is a very commonly used method, and in this has single-stranded DNA templates of the DNA fragments using that are manufactured using a special phage vector. Then this is added, and you have what do you call it? The aliquots done, and then the template DNA is added to 4 different types of reaction mixtures, including the DNA polymerase. Aliquots done, and then this dideoxy nucleotides do not have the hydroxyl group at the 3' carbon position. So this prevents a phosphodiester bond being formed basically. So what happens is, because of this, the DNA fragments terminate at the respective dideoxy nucleotide. So because of this, you will get a DNA sequence which is complementary to the single-stranded DNA template. This can be directly read on the autoradiograph gel. So fluorescent labeled primers are added, and just a second. So you have different sequences of the different types of DNA sequences basically. So like I told you, we use the dideoxy nucleotides. So these are the bases that are there, and then you get the complementary bases that are present. You have a better picture in your textbook basically. You can see. So when it comes to the techniques used for mutation sequencing, you have different kinds of techniques that are there. Denaturing the gradient gel electrophoresis is one. Then you have the chemical cleavage mismatch. So you add hydroxylamine and osmium tetroxide. This reacts with the cytosine and the thymine nucleotides. Denaturation occurs. So you can actually see the cleavage mismatch. Then you have the single-stranded conformational polymorphism. Then you have protein truncation tests. So what happens is, under appropriate gel electrophoretic conditions, you can actually see the the mutations that are present, if any. And in protein truncation test, what happens is, in this test, the cDNA produced by the RNA using the reverse transcriptase, that is RT-PCR, you get a special primer that is a promoter and translation initiator sequence. Then resulting DNA is translated using this transcription translation system. This product, whatever you get, is analyzed on the gel. And this indicates if at all there is any kind of frame shift mutation, splice site or nonsense mutation, anything present. So that's what. I think I will continue the rest of the lesson tomorrow. So it should take around 5 to 10 minutes of your time. After this, I will go ahead with the next lesson, which is the cytogenetic culture setup and harvesting. All right, I will stop right now. Okay, thank you so much.
Thank you, ma'am.
Thank you, ma'am. Thank you, ma'am.